All right. Welcome everyone to today's cryo-EM current practices webinar. You are joining the 3 national centers established by the NIH Common Fund Transformative High Resolution Cryo-Electron Microscopy Program, the Pacific Northwest Cryo-EM Center, PNCC, the Stanford SLAC Cryo-EM Center, S2C2, and the National Center for Cryo-EM Access and Training, NCCAT. My name is Christina Zamani. I'm a scientist and training liaison at NCCAT, and today I'm also your webinar moderator. The cryo-EM Current Practices webinar is an ongoing series that takes place the last Thursday of every month at this same time. We've had a growing audience since our first talk last fall, and I'm looking right now, there are already 242 people in the room right now. We've really been humbled by the size of the audience joining us, and we're really pleased that we can bring together such a large community of cryo-EM champions from across both the US and the world. In this series, we feature speakers who can highlight the methods that they're using to obtain and highlight the methods they're using to obtain and interpret the data they can collect on the high-end instrumentation that's available at the national centers. And we hope that these talks make cryo-EM methods more accessible, particularly for any of you who are just starting to use cryo-EM in your research. And for that reason, we aim to leave plenty of time for Q&A at the end of the talk so that we can dive into the details that are most useful for you. We are recording today's talk, and we will post the recording most likely early next week to our YouTube page. You can go to tinyurl.com/cryo-EM-talks. And you will find this talk later, as well as all of our previous talks. You can also find information about this webinar series, as well as general information about the Transformative High Resolution Cryo-EM Program resources at our new website, cryoemcenters.org. And I thought I would be very brave and live demo this. So let me do this. I think you should be able, hopefully you can see this as well. So this is what that website looks like. You can get information about cryo-EM centers, the work going on by teams doing curriculum development, as well as the newly established cryo-electron tomography centers. And then on the events page, hopefully this loads quickly. You can see we have information about workshops and courses at all of the different centers, and down here information about upcoming talks. So here's the registration link for this today. We don't yet have the registration link for our next talk, but it will be here as soon as we have it. And you can also find links to our previous talks. All right, that live demo worked. I'm very pleased. And let me come back to the screen. All right. So along with being posted on that site, you should, we'll also advertise the next talk with the registration link on the mailing list, the 3DEM and CCPM mailing lists, also on Twitter and email blasts from the centers. So save the date for March 25th and be on the lookout for a registration link for that next talk. Back to our agenda for today. We have representatives from the centers here. We're missing our representative from S2C2, but we'll hope he's able to join us in a couple of minutes. We do have here Ed Eng, the manager at NCCAT, and Theo Humphreys, a scientific point of contact, or SPOC, at PNCC. And they're each going to give a quick update from their respective centers. And after that, Theo is going to properly introduce today's speaker, Bobby Hollingsworth, who is joining us today from Harvard Medical School. Also with us is Lauren Halesbeck. She's the project coordinator at PNCC, and she will be moderating our Q&A after the talk. You aren't able to turn on your microphone, so all of your questions should be asked in the Q&A window. Please don't use the chat for your questions. Put them into the Q&A window, because that's, use the Q&A button at the bottom of your screen. That's where we're going to be looking for questions. Please send them in at any point during the talk. If your questions are directed to logistics or access at the centers, our panelists will answer them directly in the Q&A box. And we're planning to save most of the questions for Bobby until after his talk, but if we see something that's best addressed right away, one of the panelists will interrupt him. Also, you can see the questions other people are asking and upvote them. Given the large audience we have, it's possible we'll get more questions than we can get to by the end of the hour. So use that upvote to help us prioritize the discussion for after the talk. All right. With that, I'm going to turn over and let Ed go first.
Okay. Hello, my name is Ed. I'm the manager here at NCCAT. We offer nationwide access to 4 dedicated cryo instruments, as well as cross-training with our remote cross-training program now that most of our operations are done remotely. I want to point out we have a couple of things coming up. Due to the pandemic, we had to time shift and reconfigure some of our workshops. So our short course in tomography that was postponed is now held virtually, and it's going to be held in April. And you can find more information on nccat.nysbc.org, as well as how to sign up for access. If my colleague from S2C2 isn't here, I can also introduce S2C2.
Yeah, Ed, I'll let you do that. Take it away.
Okay, so S2C2 is housed at the SLAC National Accelerator at Stanford. They offer also at no cost access to data collection as well as cross-training. They have an in-residence and remote cross-training program very similar to how we run. They have 3 dedicated instruments, alpha, beta, and gamma. And what's interesting is that alpha has a Falcon 4, beta, gamma are GIF K3s, and they have some fringe-free illumination. So if you want to find out more about S2C2, please go to the website.
Thank you so much, Ed. And now I will turn it over to Theo.
Hey, my name is Theo Humphreys, and I work as a scientific point of contact at the Pacific Northwest cryo-EM Center. The PNCC offers 2 proposal types, including a limited access proposal for preliminary data sets up to 24 hours and a general access proposal for larger projects, which grants up to 480 hours of microscope time a year for 2 years. As far as instrumentation goes, the PNCC has 1 Arctica and 4 Titan Krioses. 2 of these Krioses are equipped with a Falcon 3 and standalone K3, and the other 2 Krioses are equipped with the Falcon 3 and K3 with bioquantum energy filter. We'll be outfitting one of these Krioses with a Falcon 4 cold phag and biocontinuum energy filter this year, and all of our Krioses will be updated with fringe-free imaging by the end of the year. For sample preparation, we also have a VitraBot and a Leica GP2, and are looking forward to the installation of a VitraJet soon. If you're interested in applying to the PNCC, applications are due every month on the 1st at 11:59 p.m. Pacific time. For these applications, we leverage the EMSL user portal, which also handles proposal tracking. In general, it takes about 40 days from the first for proposals to be reviewed and received an award letter. And once approved, there's a recommended user onboarding with our project coordinator, Lauren. On approved proposals, the PNCC offers both screening and data collections for projects focusing on single particle analysis and tomography. Additionally, each approved proposal will be delegated a scientific point of contact, such as myself, who can answer scientific questions about any part of the cryo-EM workflow. The PNCC is currently under modified operations and is offering one-on-one remote training and small remote workshops. Once we're able to have visitors on site again, we plan to offer small on-site workshops covering microscope operation and sample preparation. And if you'd like to apply or learn more, you can visit our website or reach out to us. We hope to hear from you soon.
Thank you, Theo. I'm going to stop sharing my screen so that you can introduce Bobby.
Yeah, today I have the honor of introducing our webinar speaker, Bobby Hollingsworth, who I've had the pleasure of working with at the PNCC. He received his bachelor's degrees in chemical engineering, biochemistry, and chemistry at Virginia Tech. During this time, he gained initial exposure to cryo-EM through a summer internship with the Supermanium Lab. And at Virginia Tech, he completed research with doctors Anne Brown and David Bevan, where he used molecular dynamic simulations to look at the HIV envelope and its transmembrane region. During his time in their lab, he developed a love of mentorship and open science. Bobby is currently a fourth-year graduate student at Harvard Medical School in the lab of Dr. Hao Wu. Here he is able to explore his general interests in cellular signaling, circuitry, and logic with the goal of discovering places where things go awry in disease and the means of restoring normal function. His current work studies regulation and innate immunity. In particular, they're looking at structural and cellular mechanisms modulating the activation and assembly of CARD8 and NLRP1 inflammasomes. And without further ado, I'll let Bobby take it away. Thanks.
Great, Thea. Thank you so much for that introduction, and thank you to the service centers, and in particular PNCC, for inviting me to give this talk. So today I'm going to tell a story about these inflammasomes, NLRP1 and CARD8. However, I'm going to give a more technical talk focusing on our journey to optimize the cryo-EM conditions and sample conditions, but also provide some biological context for our structural results as well. And so I hope that these insights are broadly applicable to a variety of different people pursuing many different types of cryo-EM projects. And before I begin, I'd like to thank my core collaborators on these projects, Laurent David and Humayun Sharif in our lab, as well as Andrew Griswold in Dan Bachovshin's lab at Sloan Kettering, as well as Yang Li, who's an assistant professor at UT Southwestern, all of whom I've had the wonderful opportunity to learn a lot from and really grow in these projects with them. At the end of this talk, I'm also going to post all of these slides on our Open Science Framework. Here's the link. If you miss it now, that's fine. The talk will be posted later, and this presentation will be up with embedded links and some notes in the slides in case those might be helpful for you. First, I'll give some background into innate immunity in inflammasomes, in particular the NLRP1 inflammasome, and then I'll launch into our structural determination efforts for these poorly behaving regulatory protein complexes, and then talk about helical filaments. And within each section, I'll first discuss the challenges we faced, and then I'll talk about sort of the core biological conclusions we had. At the very end, I'll just very briefly touch on these small proteins that we've started working on, in addition to different open access tools that we've started taking advantage of to really accelerate, or hopefully accelerate research in our field and in structures in general. Discriminating self from non-self is a fundamental challenge of all life. Complex organisms would not exist without having evolved these germline encoded sensors with primordial immune functions. And so human innate immunity, and in particular these cellular sentinel pathways, were shaped by an ancient evolutionary arms race against different opportunistic invaders. These innate immune sensor proteins and pathways recognize diverse danger signals, such as flagellin present on a variety of different bacteria, or RNA present on many different viruses, undergo conformational changes that lead to cellular signaling pathways that often result in cell death, as well as the release of pro-inflammatory molecules. And this allows immune cells to taxi to the site of perceived danger and deal with these perceived threats. However, these immune sensors are a double-edged sword, and the efficacy of them comes at the cost of inflammatory disease sometimes. While these sensor proteins are absolutely necessary to recognize a variety of different pathogenic insults and take care of them, they also can be aberrantly regulated. For example, when non-pathogenic insults like uric acid crystals in gout are persistently present, or when these inflammasomes and other sensors have germline mutations that lead to their persistent activation, they can result in damaging inflammation. And that can cause a variety of different diseases. And so understanding the repertoire of different sensor proteins, their precise functions, mechanisms, and structures can be helpful for designing medicines for a spectrum of diseases, whether it be pathogens that have evolved to circumvent these regulatory mechanisms, or diseases mediated by these inflammasomes themselves that might cause cancers and autoinflammatory diseases. And so these different inflammasome sensors and sensor proteins in innate immunity recognize different danger signals. The two that I'm going to be talking about today, NLRP1 and CARD8, recognize pathogenic effectors like proteases and other danger signals. But in contrast, for example, the NAPE NLRC4 inflammasome recognizes flagellin and rod needle proteins. And so due to the need to recognize these different pathogenic insults, these different inflammasomes are expressed in different cell types, undergo different regulation, and have different levels of control at the structural level as well. So they're uniquely fascinating to study from a structural point of view. For example, the NAPE NLRC4 inflammasome, these NAPE sensor proteins bind their cognate ligands, undergo conformational changes that leads to the recruitment of NLRC4, its oligomerization, and then this platform can then recruit downstream signaling effectors and adapters. And so this was some beautiful cryo-EM work done by Liman Zhang in our lab, now at OHSU, as well as others in Gigi Chai's lab, Eva Nogales, and Russell Vance's lab. And this methods paper is particularly helpful for authors elaborated on some of their structure journey as well. And so this really just goes to show that cryo-EM has been a very powerful technique to study different innate immune sensors, particularly because of their massive size, sometimes in the kilodalton or the megadalton range, also their heterogeneous composition. In contrast to that, I'm going to discuss briefly the activation mechanism for NLRP1, and this will be important later in the talk, where we discuss interpreting the structure in the context of the pathway. So NLRP1 and the related sensor CARD8 contain this autoproteolytic function defined domain, or FIND, I've abbreviated here. Autoproteolytic activity in that domain splits FIND into two non-covalently associated subdomains called ZU5 and UPA. And these two halves of the protein after autoprocessing remain self-associated, and physical separation of the C terminus, which is the business end of the protein, is required for its activation. That's accomplished through N-terminal degradation by the proteasome. A variety of different activation signals can ship NLRP1 to the proteasome, where it's degraded, but because of this break in the polypeptide chain of the function-defined domain, as NLRP1 is being degraded by the proteasome, the C-terminal portion can essentially just disassociate, oligomerize, and form an inflammasome. And so this allows NLRP1 to respond to a variety of different pathogenic effectors that destabilize it or try to degrade NLRP1. However, like these other inflammasomes, NLRP1 is a double-edged sword. And so mutations in NLRP1 can result in a variety of severe germline-associated diseases in humans, many of which are in these N-terminal regulatory regions of the protein. And they are generally presenting as keratoacanthomas, which are a variety of skin-associated lesions and recurrent cancers because of the very high expression of NLRP1 in the skin and airway epithelia. However, I'd like to draw your attention to this particular mutation in the C terminus. And this was particularly interesting because it was recently found to result in loss of interaction with a repressor protein called DPP9. DPP9 is shown to directly bind NLRP1, and genetic ablation of DPP9 also activates NLRP1. And recently, these authors have shown that mutations in DPP9 also lead to autoinflammatory diseases. And so DPP9 is a bona fide repressor of NLRP1 that binds it directly. And so the question became, how does DPP9 binding repress the NLRP1 inflammasome? And we decided to look at this through a structure lens where we hoped that the structure would guide mechanistic hypotheses that we could then go and test in cells. And so now that I've sort of set the stage for the microscopy, I'm going to go into our sample optimization and protein preparation steps all the way through, the cryo-EM optimization, and I'll include this little diagram on the left to sort of break this into discrete steps from sample preparation, grid optimization, collection and processing. However, of course, this isn't necessarily math, but I'll try to keep it to that as best as possible. So the first challenge we had was in expressing the protein. NLRP1 was very aggregation prone. It expressed poorly in all expression systems except when co-expressed with DPP9 in XB293F mammalian cells. And so we weren't able to purify this separately for biochemical reconstitution experiments. However, we were able to get enough protein for making grids from this mammalian cell system. And I immediately noticed that the full-length protein would migrate to the carbon and was pretty aggregated in cryo-EM grids. And so I decided to truncate these two self-oligomerization prone domains called pyrin and card. And so these are known self-association domains, and fortunately for us, didn't make up the DPP9 binding interface. And upon doing that, the grids looked much better and the protein was better behaved, expressed much more highly as well. And so with this truncated protein co-expressed with DPP9, we could move on to optimizing the purification. And we ran into an issue here where the protein was apparently sticking to column support in size exclusion chromatography. So here what I'm showing are FPLC traces following a flag pull down. I run mono-Q anion exchange. And here on the y-axis is the absorbance at 280 and the elution volume on the x. And I get this very nice peak, separate out any non-specifically bound proteins. However, when I take this protein and put it directly onto a size exclusion column, it almost entirely sticks to the column support. The peak back here is just a nucleotide peak. And essentially the protein never comes off of the column. It's either getting stuck in the filter or on the column support itself. And so this was throwing us for a loop for some time, but I just went back and did negative stain of this peak and it looked gorgeous. Not introducing a sizing step maybe introduces sample heterogeneity, but that can be sorted out at the classification stage. And so we went forward with just dialyzing this peak directly and making grids. A lot of people in our lab have noticed this problem, particularly with membrane proteins or if they have low input. And so a lot of people have taken to using gradient ultracentrifugation in our lab, followed by desalting and then immediately making grids. So now we have an optimized sample,
Bobby.
reproducible protein prep. Yeah.
Bobby, did have a question
Sure.
about the EXPI 293F cell line. They wanted to know if you use that cell line specifically and if you could comment on that system as a transient transfection.
Yeah, I really like this mammalian cell system for this particular protein compared to bacteria and insect cells. It just produced so much more. I think that our lab in general has taken to using it for a lot of different projects. Compared to plated hex cells, they're a lot easier to manage in terms of a expression system because everything's in suspension culture. The drawback is that the media is very expensive, and so optimizing the sample and being very careful about that during initial purification steps is really important. But I think, did that answer the question?
I think so for the transient transfections. So they were also curious if you had to use the cell line specifically, or if there was other cell lines you could have used.
Yeah, so none of the non-mammalian cell systems worked for us. I'd never tried plated hex cell systems or HeLa cells, for example. And so I don't know if those would be valid purification systems for this particular protein. But for transient expression, they express very well. In our paper in protocols.io for this, we talk about how we boost expression by adding glucose and VPA after transient transfection. And a lot of people in the lab have taken to using the BAC-MAM system as well. And that oftentimes has even higher yields than transient transfection.
And then one final question is, why did you dialysize after the anion exchange?
That's a good question. The buffer is pretty high salt, first off, and so wanted to reduce it down in order to have less background potentially on our EM grids. And then also I use a tris buffer for anion exchange. It's just a little bit better in the buffering range. And so later when I talk about cross-linking, it's important to get rid of the tris buffer for using glutaraldehyde cross-linking.
Awesome, that's all I have so far. Another question for the end, but we'll wait.
So next, so now that we have this prep that's reproducible, we decided to start screening on cryo-EM grids. And we were noticing from our small data sets that we were getting this dissociation in grids that I just alluded to, where most of our particles were aligning to these empty DPP9 particles that were missing in LRP1. We were losing about 80% of our data due to that. And this is probably just due to dissociation at the air-water interface because the protein complex purified together. Stoichiometry looked very good by SES Page. And so the solution here was to cross-link with glutaraldehyde. And we did this very simply. We just simply mixed the protein with glutaraldehyde for 5 to 10 minutes immediately before plunging our cryo-EM grids. We'll make a grid or two and then proceed to data collection. And that shifted the population of particles to almost 90% complexes. So we're recovering a lot more data from each micrograph. However, that's still presented with a severe orientation bias, of course. And so we had DPP9 situating on the grids in this particular orientation where we weren't getting these top or bottom views. And so that leads to some really big issues with the EM density itself, where the density is anisotropic. It appears almost visibly like perforated with holes like this Swiss cheese, and the density is all stretchy. And so it's impossible to build a model from this. And so, we had to turn to many different techniques. We tried different grid types, additives, and what ended up working for us was just essentially brute forcing this problem by tilting the stage to distribute these orientations over a new cone. And so what we did was pipe in the angle distribution file generated from Reliant from refinement into the program called cryo-EF, and here's the citation for that. And cryo-EF recommended, based on this initial angle distribution and orientation bias, that we tilt the stage at 37 degrees, and that ended up working perfectly for us. And that orientation bias was reproducible across different data sets, so we didn't think it was attributed to ice thickness or anything. So for us, that particular tilt angle worked well. Some tips for tilted data collection in general is that using an untilted data set can be particularly helpful in calculating the tilt angle using a program like cryo-EF, because it's almost impossible a priori to just guess a tilt angle that's going to work well and determine whether or not the sample has an orientation bias in the first place, unless it's just so readily apparent from screening. Additionally, we use a higher electron dose and higher defocus, and I have those compiled at the very end of the presentation if anyone's interested in our particular collection schemes. Due to the thicker ice when tilting, we have to use lower particle density on our grids to avoid overlap. And so when two particles are very close together on an untilted micrograph, when the stage is tilted, these can overlap a little bit in the Z direction, and then they're essentially going to, the noise there is going to make those particles unusable for a high resolution reconstruction. And so a lot of data can be thrown out if the grids are made too densely. And so that generally requires more tilted micrographs to be collected than untilted. And tilted collection schemes are generally a little bit slower. And so there is a trade-off in data collection here and data quality when considering whether or not to use this versus other methods to help ameliorate a orientation bias. And finally, we use gold grids, particularly either gold foil grids or gold support grids, in order to reduce the drift, which is a little bit more extreme for these tilted data collection schemes. Additionally, for processing, it's very, very important to get a good initial, yeah, sure.
Sorry, more questions. So on the last slide, people are wondering why you use the tilted data set or data collection instead of tomography using M.
Yeah, so tilted data set, we can still do single particle analysis with the tilted data set. And so tomography is, of course, a valid technique. These are very small protein complexes. I don't think I mentioned it before, but our complex is approximately 300 kilodaltons in size. The viewable area, the area that's resolved, is even smaller in the end. And so I'm not entirely sure if applying tomography to the problem it probably would help, but we just chose to continue using single particle, collecting at an angle that would just give us these missing views, and then processing that data essentially in the same way.
And then also, did you combine the tilted data with the non-tilted data?
That's a great question. We had a couple of schemes that I summarized at the very end. After the acknowledgement slides that anyone can take a look at. We do for a couple of our data sets, and we don't for others. It depends on how extreme the orientation bias is. Generally, we had more tilted data sets than untilted, because if you combine the two with, say, an equal number of micrographs, you just recover orientation bias by just adding in so many particles from one view. But the untilted micrographs have a little bit more information generally. And so we tried different things, and it's worth testing both for your particular case.
Awesome, thank you.
And so for data processing, we found that a good initial estimation of the contrast transfer function is absolutely essential for further processing steps. This is because when collecting data at a tilt, the CTF is going to vary across the micrograph. So it's really important to use things like per particle CTF correction with GCTF or GoCTF, or CryoSpark's patch CTF function works actually just beautifully for this. Otherwise, alignment in initial 2D and 3D classification will be quite poor. And then once we move to refining the final structure, what we do are refinement cycles. So we run 3D refinement in Reline, followed by CTF refinement, and we run that in cycles as you get better and better approximations of a more correct contrast transfer function. And eventually that converges, the resolution plateaus, and that's when we stop running the refinement cycles. And this is described beautifully in, I think, in Dimitri's paper here, in a lot of these things as well. So I'd really encourage everybody to go read that paper first, because they did a lot of beautiful things. We got started by reading that paper. It was really fantastic information. We had tried other things as well. As I mentioned, we had tried additives. So even for soluble proteins, sometimes detergents help with a orientation preference issue that just comes with the drawback of generally requiring more protein for a higher concentration for it to go into the holes. Additionally, functionalized support and these functionalized grids are becoming more and more prevalent. There's been some beautiful work in making new grid types. However, these are oftentimes not completely accessible because many of them aren't commercially available or harder to make. But the barrier to entry isn't impossible. So if it's necessary for the project, it's worth looking into those. I'm sure they'll become more and more available as time goes on. And one really interesting thing that we noticed was that pH of the sample actually makes a big difference. And this was sort of an accidental discovery. A graduate student in our lab was preparing a protein at pH 8 normally, and he shifted the pH to 7 to just try some changes in the sample condition, and went from a reproducible, just terrible orientation bias to recovering all of the views of the protein. And that just could be due to different surface properties of the protein at different pH that then lead to different interactions with the air-water interface. And that's something really simple to definitely take into account and maybe try on certain projects. In terms of data processing, that was the last hurdle for us. And I won't talk too much about the 3D classification schemes. Humayun did a fantastic job, processing the data set at a later stage. However, we also encountered issues with particle picking. And so for our 2D based picking in Relion, despite everything we tried, it seemed like we were getting a little bit of our picks off-centered. We weren't recovering an amazing amount of particles from a data set of about 6,000 micrographs. However, when we trained a cryolo model, I manually picked 10 micrographs for this. After a single round of classification, we were having 1.8 million particles with beautiful alignment. And so it's worth going and spending a good amount of time in the particle picking stage, because ultimately the whole data processing scheme is just refining the particle stack. And so Cryolo has been great in our hands. I know some people have been using Topaz a lot more and other particle pickers. Just worth trying to just get the most out of a data set. And so I guess if we don't have any questions, I'll move on to briefly discussing the structure findings. That's basically all the technical hurdles we encountered in this project. And so I'll just tell you a little bit about what we learned. And so we found that 2 copies of NLRP1 bind each DPP9 subunit, which is a dimer. So here I've grayed out one half of the dimer. And we only resolved the function-defined domain of NLRP1, which was the interaction domain with DPP9, and the rest probably averaged out during our data processing due to these flexible links between these upstream and downstream domains to the function-defined domain. What was pretty interesting was there were 2 of these copies, but both copies weren't the same. So the first function-defined domain that we call function-defined domain A, find A, had a zoo5 domain that's present on the N-terminal end of the function-defined domain, and the UPA that's present C-terminus to the cleavage site. However, for the second function-defined domain, we only saw UPA on the C-terminus. And so we had wondered, what happened to the second zoo5 domain. And so what I did was take this pair from the first site and just rigidly superimpose, essentially just basic, PyMol, take this, superimpose it onto the second UPA and bring a theoretical zoo5 with it. And we showed that zoo5 would clash with all of these other protomers in the structure and really wouldn't be reasonably accommodated. And so we wondered where it went, was it dissociated, degraded? And going back to the biology of the pathway, we knew that N-terminal degradation would lead to the degradation of the ZOO5 and the rest of the N-terminus of NLRP1 and release these C-terminal fragments that basically only contain UPA and then the C-terminal CARD domain that we don't see in the structure. And so, I think us and a lot of people had thought this was really only occurring under potentially pathogenic conditions, but we're not perturbing the cells in any way here other than overexpression. And so perhaps this is a mechanism to soak up these inflammatory fragments during just normal protein turnover in order for them to not activate the inflammasome due to that or small perturbations to homeostasis. And so going back and interpreting this in the context of our structure using this cartoon, where I'm just graying out one of the DPP9 subunits, this is sort of a ternary complex with three different components. DPP9, full-length NLRP1 at this A site, which requires an UPA and ZOO5 to bind this site, as well as a already degraded NLRP1 binding this second site here, site B. And so what this allows is this complex to only capture the C-terminal fragments when there's full-length NLRP1 present in the cell. There's cooperative binding, so neither of the halves can bind without the other. And we went on and did some structure-guided mutagenesis. I won't go into any of that data here because of time, but we looked at these different binding interfaces and reconstituted these in cells. Andrew Griswold did some amazing work there to show that if you break up this complex, it results in inflammasome activation and signaling. And if you reconstitute the complex so that these C-terminal fragments are soaked up, it prevents inflammasome activation. Of note, for later in the talk, there's this interesting sort of dimerization or oligomerization interface between two different NLRP1s that'll come up again later. And so we could go revisit this mechanism. We could interpret DPP9 now in the context of the pathway, where DPP9 isn't necessarily preventing N-terminal degradation of NLRP1 by binding it, but instead soaking up these C-terminal fragments as they're generated by proteasomal degradation, thereby sequestering them and preventing them from forming an inflammasome with low levels of protein degradation. We show that essentially this mechanism is acting like a rheostat where DPP9 is helping the cell sense the balance of full-length NLRP1, the repressed state, versus the proteasomal activated C-terminal fragments. And upon undergoing protein degradation, the pool of these full-length proteins is depleted, the pool of the C-terminal proteins is increased, and when that reaches some sort of threshold set by this rheostat, the inflammasome can form. And patient mutations result in basically skewing this ratio at all times. A lot of these patient mutations destabilize the protein. They were shown before to unfold regions of the protein. And so these likely lead to faster degradation of NLRP1 just under resting conditions. And so that leads to persistent inflammation that causes diseases in these patients. And any strategy to ameliorate this disease would have to push that balance back. And so this work isn't quite out yet, but there's some beautiful stuff by Andrew. I just told you the conclusions. I didn't really show you. And so if you'd like to look at those, all of the preprints are out. And we did some similar work as well with the cardiac inflammasome, which is highly similar. And so now I'm going to pivot to sort of the other end of the pathway and talk about a cryo-EM structure project involving different helical filaments. And so upon activation, inflammasome sensors undergo conformational changes that lead to their assembly into supramolecular helical filaments mediated by CARD domains and or pyrin domains. And these filaments serve as signal amplification platforms to recruit downstream adapters and effectors between CARD-CARD and pyrin-pyrin interactions. And so this process has been well studied by our lab and a number of others. However, we were interested in this case for NLRP1 and for CARD8 in that they contain this UPA domain that I described previously and wondered if it played a sort of architectural role in the assembly of these inflammasomes. So to solve the structure of these filaments, we used a protein expression method that's a favorite in our lab for purifying filaments. And that is to purify first a soluble fusion protein that's monomeric or slightly oligomeric from E. coli to homogeneity, and then cleave the fusion tag in order to initiate filament formation in vitro. And then we can control these cleavage conditions in order to get good protein for cryo-EM grids. And so this requires optimizing the construct itself, the choice of linker and fusion protein in order to sterically hinder the polymerization, in this case of the CARD domain. Optimizing in vitro the cleavage reaction requires optimizing things like the protease concentration, the sample concentration, temperature and time, and whether or not to refold, which is a condition some people use is refolding protein to initiate filament formation. And to achieve, both complete cleavage, elongated filaments, as well as avoiding aggregation, which is a big problem with these. So for us, after optimizing these cleavage conditions, we would put the cleaved protein onto size exclusion to purify out remaining proteases and the tag, and we'd recover the filaments in the void fraction of a size exclusion column. And we found that despite our best efforts, we would never get complete cleavage of the MVP tag, which is what we used to purify this protein. And these MVP tagged are incorporating into our filaments and probably preventing their elongation. And so we were getting smaller filaments than would have previously been observed for card filaments, probably 5 to 10 times shorter. And so they're a little stockier on these cryo-EM grids. And we hoped that the flexible tag would average out during processing. And that was luckily the case. And that we could have a reconstruction through segmented particles, even with these small filaments, and luckily we could. Of course, we ran into grid optimization issues with these filaments as well. In particular, the NLRP1 filament would not go into quantifoil copper grids for some reason. It would stick to the carbon. It would, stick around the edges, and everything inside the holes was pretty aggregated. However, Laurent David in our grid, in our lab, has noted that sometimes filaments just behave better in C-flat grids. We don't know why. And so we used C-flat grids, and it just worked. And so it's just worth trying different grid types, because these small differences in surface properties of these grids just may make a world of difference when it comes to a cryo-EM project. I'm not going to talk about helical processing too much, other to mention a couple words of caution, like that during data processing, a lot of local minima for the helical parameters can be achieved. And that leads to really artifactual structures. And so it's really important to inspect structural features when imposing helical symmetry. For our own processing, we use start-end picking. And so this is manually going and picking the beginning and end of all the filaments. And for our project in particular, that was important because these filaments were quite heterogeneous. They were small in size and sometimes overlapping. So that helped us curate our data set as best as possible. It only took a day or two and only required about 1,000 to 2,000 micrographs for a high resolution reconstruction because of the helical symmetry. Recently, there have been these other algorithms implemented in Cryolo and CryoSpark for automated picking, and so that probably works pretty well with straight filaments. Additionally, for initial 3D structures, that can sort of be really defining in the data processing scheme. We use Relion to generate a simulated helical lattice, which requires a good estimate of the helical parameters. Essentially, it's blobs of density around a simulated helix where you give it the rise and twist. However, these sort of just featureless cylinders are also a good way to start. So here's kind of a picture of one, just essentially a tube of density. And that comes with the problem that when refining the helical parameters, you might not end up with the correct helical parameter. So it's important to consider these things when going through a project. CryoSpark also can generate initial models with ab initio reconstruction, but that doesn't work in all cases, particularly when we're only seeing one view of the filament. And so our processing scheme, devised mostly by Yang, was to pick and segment the particles using a generous box size of like 800 angstroms, doing 2D classification and varying the parameters until the filament's situated very straight in the box. Then generate an initial 3D model like on the previous slide. And we refine that 3D model against the best single 2D class in order to refine the helical parameters. And we low pass filter that new model and use it for that new density and use it for 3D classification with all of the good 2D classes. Then finally, we pick several of these 3D classes that look good, do refinement cycles with particle polishing and CTF refinement until we get the best structure that we can achieve. And so I'll tell you a little bit about the results. We were a little bit sad to find that we only resolved the central card domain of these filaments. And we posited that perhaps the UPA subdomains were flexibly linked, like on this cartoon here, around the periphery and either weren't following helical symmetry or, despite not imposing symmetry and other tricks we tried to play, were just heterogeneous. And so what was immediately interesting about these two filaments were that NLRP1 had a dimer. Had a central helical filament flanked by additional card dimers. And I won't talk about this here, but we discuss it in in our paper, and that's back to back with one of our colleagues' paper. So it's worth mentioning. They had some fantastic results as well. So we wanted to get at the role of the UPA subdomain using biochemical and cellular techniques to complement the structure, or perhaps even look back at our previous structures. And when we were expressing just the card domain alone, we immediately noticed that very high concentrations of card domain were required for initiating filament formation. However, and for NLRP1 at least, it was just not attainable with how high we could concentrate the protein without it aggregating. However, when we fused the UPA domain back, we had a construct representing what the inflammasome would look like in UPA and card domain. We found that much lower critical concentrations of the protein were required in order to nucleate these filaments. And so the UPA itself is playing a role in enhancing and promoting the assembly of these filaments. And Andrew and Elizabeth in Dan Bachovic's lab then went and showed this holds true in cells as well. And so we were wondering how it was doing this. We tried to express these alone, and we found that these UPA domains in isolation would form small oligomers or little aggregates. And looking back at our DPP9 complex structures, we saw that there was this UPA oligomerization interface that I mentioned previously. And if we just modeled that interface out, we could generate these small polymers of UPA subdomains that could be oligomerizing. And so we hypothesized then, or I guess concluded, that these UPA domains, by self-associating, are avidly promoting the assembly of this central card helix. And so without these UPA subdomains to self-associate, the critical concentration required to form a card oligomer is much higher. To tie both ends of the story together, we found that this DPP9 ternary complex is comprised of a full-length NLRP1 and this C-terminal fragment that contains an UPA subdomain and a card subdomain. Because it's in this complex, that UPA card fragment is sterically hindered from forming any sorts of polymers. Upon degradation of full-length NLRP1, these C-terminal fragments are free to self-associate. The UPA domains cluster and allow the card domain to form a central card helix. And that's all I have for this part of the talk. I just wanted to mention briefly some efforts to resolve small proteins in our lab. And so this has become routine for us for very highly symmetric or rigid particles. However, when there's flexibility, particularly flexibility that isn't in discrete states, like conformational continuums, like in this protein that Kelly's been working with, where the protein rotates freely about this hinge domain, it's been a real challenge to resolve those structures because density particle subtraction is often not producing a sufficient amount of remaining density for particle alignment. And so we found that screening buffers for thermostability, so this is sort of taking a play of the crystallographer's toolbook and using thermoshift assays to optimize buffer components. In addition, finding small molecules or nanobodies that stabilize a particular conformation is really useful. And that's what Kelly did here. She uses a small molecule to stabilize her protein and is able to see this density and is currently working on that project. And this is broadly applicable to every cryo-EM project in general. I think Zod talks about this in his talk at this forum a couple of months ago, where he finds a small molecule that can stabilize a transmembrane region of a protein. And so that can be a very useful tool for resolving protein structures. I'll conclude by talking about some open access tools that we become more and more active with. The first being AddGene, where we can deposit our plasmids. And I encourage everyone to deposit their expression constructs as well as functional constructs. And the folks at AddGene are wonderful, and they distribute these super efficiently. So it really helps accelerate research. Protocols.io is a tool to deposit extended protein purification protocol, where you can publish intermediate steps and pictures of things that you might not include in a typical paper, like pictures of STS page gels at all stages or little diagrams and things that are very helpful for other biochemists to replicate the purification. And Empire just opened up to publish or to deposit the raw cryo-EM data from data sets. And I saw this quote circulating around Twitter recently about molecular modeling, where essentially a molecular modeling project can go on forever before depositing the coordinates. And sort of the same holds true for working with cryo-EM density. And in the future, there are going to be better algorithms for processing this data. And so it's worth, depositing these really good data sets. And in the future, they could be mined for more information or used just as case studies for tutorials and workshops. Finally, I've been publishing just depositing PyML session files on Open Science Framework, which is effectively just a data archiving tool. And so for every figure that goes in the paper, put an associated PyML session file in Open Science Framework. So if anyone's interested in playing around with that particular view that they see in the paper, they can just follow the link that we embed in the, in the method section, and then just quickly download it. Everything's, colored very well. Saves people that are used to working in PyML some time, and is especially helpful for general biologists who perhaps don't have a ton of PyML experience. Before I go into acknowledgements, I wanted to say at the end of the slides, if anyone's interested in downloading these, I've compiled all of the tips and tricks for each of the cryo-EM structures that we've worked on that are published, or some that are coming up soon. And so some of these links may or may not be live yet for the data. But at least I'm including the tricks to solve those structures, in addition to the data processing and collection schemes. And so with that, I'd like to thank wonderful colleagues who we've talked to and published back-to-back with, in addition to wonderful collaborations within the lab, outside of the lab, where I've learned a ton. Amazing time for doing a PhD in structural biology. These service centers and HMS's cryo-EM facility opening up right now is really instrumental to moving these projects forward, the pun slightly intended. And the expertise of the facilities, people at the facilities, has been just incredible to learn from them. Just wanted to point out that Richard, Sarah, Sean, Theo, Janet, and Ed have all been super awesome to collaborate with. And so with that, I'll take any remaining questions. Thank you.
Hi, Bobby. So we got quite a few questions, and I've tried to put them all together in a way that makes sense. But the first one is, can the inflammasome be drugged for a novel therapy?
Oh, absolutely. And the startup space there is incredible. There are a ton of companies that have been launching to try to directly drug these inflammasomes. And so it's absolutely a drug target.
Perfect. And then another popular question is, have you ever tried graphics, glycerol gradient, and glutahydramine gradient to purify protein complex?
Yeah, this is a great question. Craig actually asked this too in a practice talk we had this week. And we actually didn't for our project because we found that it was really, it was a simple way to check if cross-linking might be useful to us. And it happened to work perfectly when we cross-linked it immediately before plunging the grids. But graphics is a fantastic technique. Laurent, who's on some of these projects, has used it in her projects previously. And so trying the two is definitely a good idea for dissociation problems. However, it's just much easier to just mix the glutaraldehyde with the protein immediately before plunging grids to get some idea if that'll be helpful. And of course, when we do this the first couple times as well, we're monitoring it on SDS page gels to see if we're getting covalent interactions then between the two different proteins.
Awesome. And then in a similar line, do you think that grid preparation optimization, the blotting time, whole size, protein concentration, or additives could have worked instead of cross-linking the glutaraldehyde?
Potentially, I think that particularly optimizing additives and grid type would maybe have helped with the dissociation issue, because just varying the either the properties of the protein itself or interactions with the air-water interface by trying a different grid type might just completely alleviate that issue. And so it's definitely worth trying pretty much everything, kind of throw the kitchen sink at it in a logical fashion as you're going through the project.
And then they're also wondering, how extensively did you have to optimize that glutaraldehyde cross-linking conditions? And have you seen it cause a lot of aggregation in the sample?
First part, how extensively, for us, we were just really lucky in that just incubating for 5 to 15 minutes. How we generally do this is, we mix the protein and glutaraldehyde in a PCR tube, put it on ice, plunge the grids as soon as, the ethane is ready. And so we'll probably get like a 5-minute time point, and then we have another time point somewhere between 10 to 15 minutes, and we record everything very carefully. And generally, when we make those duplicate grids, at least one of them is nice and doesn't have aggregation. However, with another project I'm working on, that one that you saw, the Kelly's protein there, she was having some issues with glutaraldehyde cross-linking where there was a ton of aggregation. And so it's totally protein sample dependent, probably buffer component dependent as well. And so what we've done in other projects as well is varied the concentration of glutaraldehyde, dropped it down a little bit, or these other cross-linking methods that were mentioned earlier can be helpful.
They're also curious for what your cross-linker concentration that you use that worked.
Yeah, for us, it was a final concentration of 0.02%. And so we prepare a tube with 0.2%, 1 microliter in the tube, 9 microliters of protein, and then just incubate on ice.
And then how big was the DPP9 NLRP1 complex in kilodaltons?
Kilodaltons. So the resolved portion of the protein was about 260 kilodaltons. If we include all the domains that averaged out, it was somewhere in the 420 kilodalton range, I believe, or even larger, like 520.
Okay. There was a specific question.
I'm going to interrupt for just a second because we're coming up on the hour. So I think anyone who's got another meeting to head off to will do that. And we have many unanswered questions. So we'll stick around for a couple minutes as long as Bobby can manage. But I think even then we won't get to everything. I will forward the questions along to Bobby. And he did mention the slides will be available and the recording will be available as well, including the time we go over a little bit. But thank you for everyone coming. These are really fantastic questions. I think Bobby went over like so many of the bottlenecks people have with sample prep and data processing. It's a bit of a whirlwind. And yep, find Bobby's resources that he told you about. Find the talk on our website. And I'll let you get back to it.
Yeah, I'm happy to stick around for as long as people have as well. I don't have any immediate engagements, and so I can just keep going as long as people are patient enough to keep asking me the questions.
So the next question that I have is one that was slide specific that I wasn't able to get in. But what is the purity in peak 2 of ion exchange? I think this was like slide 15. It looks much pure in SD200.
Yeah, so because, that's a good point in terms of number of bands. I don't have them labeled here. I have the bands just for crowding. I have them labeled in the sort of the slide guide. And so this protein, it has autoproteolytic activity. So full-length NLRP1 comes out as 2 bands, the top 2. Full-length NLRP1, autoprocessed N-terminus, and there's an autoprocessed C-terminal band way down the gel, and then DPP9 right here in the middle. And so we do purify DPP9 very well in the next step by, just totally getting rid of NLRP1. It really sticks to the column. And so that's what you're seeing here.
Awesome. And we're going back to the tilted data. So I know this data was collected with automatic tilt, but do you know how many images you collected about that were tilted?
Yeah, so we had a bunch of different data collection schemes. I think for one structure that went to 4.2, we only had something like 1,500 tilted micrographs. That one I think was something along the lines of, so yeah, here, 37-degree tilts, we're seeing 2,000 micrographs, 2,500. This one had a huge amount of tilt, but that structure was even better. I mean, it was fantastic. And this one was just tilted micrographs alone at about 4,000. And so when we combined, we were combining about 2,000, 2,500 micrographs at the tilt.
Awesome. Next question about tilted is how do we Use flattened maps from untilted data to guess the best tilt angle for a tilted data collection.
Yeah, it's a great question. So what we did was we just took the angle distribution file that comes out of Relion oftentimes. I think CryoSpark also has it. It's like .angdist or something. And then use this program, cryo-EF. We just pipe that angle distribution file from refining the structure right into cryo-EF, and it just estimates an ideal tilt angle, assuming that the conditions are exactly the same, of course, because if ice thickness makes a big difference, or if something was weird about that particular collection, then of course it might be different. But for us, across several data sets, it was giving us 37 degrees, and so we're confident using that tilt angle.
All right, moving on from the tilted. So more on the tilted, they wanted to clarify, did you pull the raw micrographs from the tilted and untilted data particle pick, then go for refinement?
Ooh, another great question, because we did process these in totally separate optics groups in Relion until we had our final particle stacks. We even did the 3D refinement cycles of the tilted micrographs by themselves, totally by themselves, before combining the particle stacks, where all of the particle polishing and CTF refinement for both particle stacks was done totally independently, combined them, and then did one final 3D refinement. And that was the structure we reported.
Awesome. They also said great talk. The next question is it possible to generate a lower resolution model by throwing away some particles at overrepresented orientations?
Yeah, that's absolutely a way to do it. I've seen some people do 2D classification, where they do 2D classification and then do further 2D classification of just individual views in order to get really high resolution particle stacks of those views. I think in Zod's talk, he mentioned that people just throw away particles in using 3D to avoid human bias when picking some of the 2D classification results. And throwing away particles is something I think Yang has used in his processing previously in the lab, where you can just, like whoever asked the question mentioned, you just essentially throw away overrepresentative views. In our case, that wouldn't work because the views were just so underrepresented based on the particle distribution, and so we could never obtain a high resolution structure from a balanced particle set.
Going into particle picking, does the picking of long filaments work the same way? As I see most pickers give you options of round shape.
Yeah, so so we do, if you do the middle picking like in Relion, I'm pretty confident that it's, at least for helical filaments, that all particle picking manually would work fairly the same, where you pick the start and end of a straight segment that has non-overlapping particles. However, for these other pickers, I'm not entirely sure. I've never tried myself the Cryolo picker for filaments or the new implementation in CryoSpark. So I'm not confident in how those work, but I did embed the links to those tutorials here if whoever's interested in looking at those.
Additionally, for particle picking, so they want to know if there's a recommendation for a software that could be used to pick non-straight filaments and also prevent the filaments from overlapping with each other on the grids.
Yeah, I think the best software is unfortunately yourself for that case. And so for us, we couldn't prevent overlap. There was just some aggregate. I mean, these filaments are just they have all of these oligomerization interfaces. And so, they have oligomerization interfaces that are, pointing outwards in the filaments, and they can sort of cross-link themselves. And so it's really hard to get monodispersity in a lot of different helical samples. And so because of that, oftentimes it could just mean acquiring a larger data set and then just picking yourself. And so for us, we were fortunate. It was only about 200 micrographs for these. As only, we sat down for a day, picked it all, and it was done with. But it's worth seeing if that would work for the case. So, 1 day data collection, manually picking it, you only sacrifice a day there. Otherwise, I think have to go back to the sample prep as it was mentioned and try some things. We were fortunate we didn't have to try some things.
Next question was, is it right that instead of the full-length construct, the recombinant filaments do not stick to the SEC column? Do you have an idea for why?
So in both cases, it sticks, at least for the complex structure. For the recombinant filaments, yeah, as you pointed out, the recombinant filaments don't stick, but the protein complex sticks. So we think the protein complex sticks because it contains the function-defined domain, and that's what's mediating interaction with the column support. Because we purified different truncation products of the protein, we purify most of their domains alone or combinations thereof. In the filament itself, there's just the UPA subdomain and the card domain that doesn't stick to the support. So it's probably the full function-defined domain is what's mediating these interactions. But we can't be 100% sure, but that's what we think, just based on our experience with the protein.
And then a question about helical processing. How did you determine your helical parameters or judge if the rely on output helical parameters are correct? And then how did you know how many asymmetric units were in your helical filament?
So for us, to determine, I guess, asymmetric unit in terms of, I guess, box size was sort of empirical, how big we were going to bring the box. However, helical parameters, we fortunately had a great idea of what those parameters might be ahead of time because of card domains having very, very well documented helical parameters. So we used those as a starting place. Yang actually went and did some, I think, Fourier-Bessel indexing. And so there's some fantastic papers that discuss that in length. So from 2D classes of filaments, you can derive all of these helical parameters, or at least an initial guess of what they should be. I'm not an expert there, but I'd recommend reading about those. And so for, at least in our box size, for number of subunits that end up being in the structure, I'm not entirely sure if that's what the question means, or if it's, number of subunits per turn. We just empirically varied the box size in order to get, we got better alignment with a much bigger box. And then we could fit our card domain, monomer structures that have, crystal structures to that density, and then, build it.
Next question we have, is it a good idea to increase the molecular weight of protein with bigger tag like MBP to do cryo, I'm not familiar with this, EXPT?
Cryo-EXPT, I'm not familiar with cryo-EXPT. I think that increasing the size can sometimes work very well. We've had some projects in the lab where people create sort of artificial dimers of a protein that's normally monomeric. And they know that they're engineering this, but they do it in order to get this high resolution signal, better alignment. With the tag, if it's flexibly linked, a lot of times the tag just will average out during image reconstruction anyways. And so varying the linker is really important if you want to use that additional density as something that will help the alignment. And that can help. It's just totally case dependent.
Next question was 3C protease with overnight cleavage. Did you also check with the other proteins to check the filament formation as a control? Without MPB, whether do you observe these filaments formations as well?
Yeah, so in certain products we're actually trying to purify some protein complexes from mammalian cells. And this was just a really funny happenstance where somebody was trying to purify, this was after the projects were done, card8 in one case from mammalian cells, the full length, and ran the ion exchange and then, just negative stain. And we had these beautiful filaments. It was like, it just happened that because we overexpressed it so high, it was breaking this DPP9 sort of binding threshold, checkpoint, and we could get these beautiful filaments without MBP from that prep. And then we went and just played around with that and like solved a structure, but it was, you know, too late for other project, and they look very similar. It was kind of funny. It was totally accidental and just worked the first time, which was like, you know, that feeling when that happens. But, you know, we had already finished this project.
That's lucky. Another popular question is, how do you know whether you see orientational bias because the missing views are really not there, or because they're simply difficult to pick?
Ooh, that's great. That's why spending time picking for a very long time and trying many different schemes, so important. And so at least with these, we had some higher contrast micrographs that I was hand picking. And so hopefully the cryo model that I picked, where I just, you know, spent like a day just picking 10 micrographs, was hopefully trained well, where, you know, and of course that may or may not have happened. I think Topaz has some really great workshops on how they can capture some of these underrepresented views that may not get picked efficiently because you need to pick them to train a model. And so if they're underrepresented, you might even underrepresent them through automatic picking. And so those methods are helpful. And I think it's worth it to definitely be very exhaustive in that before trying these other methods, because sometimes a data set is totally fine. But like you mentioned, you know, you just have to do due diligence in picking. And so we tried this for a very long time. So hopefully we weren't missing it. If someone goes into the Empire and then just does it, I'll be happy. I'll be happy that they can do it. I would love to see that. So hopefully we didn't just miss it.
And then somebody has a suggestion, or they'd like a suggestion from you. Is it possible to collect data of about 30 kilodalton protein, which exists as a trimer in high concentration?
Oh yeah, I mean, like 90 kilodaltons of density resolved, plus there's some symmetry. I think that's totally possible. The small protein I showed was, you know, 80 kilodaltons, highly flexible, C1 symmetric. And so I think that's absolutely possible, as long as, of course, these other things, like it's a good sample prep, stable protein, and the orientations on the grid are, you know, sufficient to have the reconstruction. I think that's totally possible. It's always worth trying, you know, finding compounds or maybe nanobodies to increase the either stability or the size of the complex. But, you know, I would say, of course, go for it.
And then another popular question was, they would also like to know how you guess the helical parameters for the filament reconstruction. They know that you mentioned the rely on helical reconstruction paper through the power spectra, but they're struggling with that. So how apparent is it for the filament sample? Did you encounter any filament heterogeneity or heterogeneity? That's it. Thanks for the awesome talk.
Yes, I guess. So for the at least for determining the helical parameters using indexing, Yang had taken the very best 2D class in order to, that was very well aligned in 2D classification. So it's worth testing different 2D classification parameters like the Z offset and the angular search in order to get the filaments to align very, very straight in the box. And so once you have these very straight filaments in 2D classes, and I can amend this to put maybe 2 pictures of when they weren't aligned and when they were before I post it. When they're very, very straight, it's much easier to do the indexing. Of course, heterogeneity can be a problem. I would reach out. Yang gave me permission to say, go talk to him. He's really an expert at this. And if you're struggling, he may know, how to work around the indexing problem in your particular case.
Okay, and then we still have quite a few questions if you still want to answer them, or we could end.
Yeah, sure.
Christina?
Yeah, up to you guys. If you want to stay, we'll keep recording. We can also, Bobby, send you the questions if you're willing to address them and put them with your slides that you're gonna make available anyway. We can also do it that way.
Yeah, I mean, we can do both. There's still a lot of people on, so I mean, I don't mind staying. It's totally up to y'all's time. I mean, I don't want to impose on anybody.
Let's go 5 more minutes, and at 1:20 we'll take the rest of the questions that aren't answered, and you can manage those by typing, if that's okay with you.
Perfect. Yeah.
So then why is it important to get horizontal 2D class averages and a 2D classification of helical filaments? And then also your paper mentions that shift of 6 angstroms for each segment box. Which box and rely on have you put this value?
Which box for shift? And so for helical shift, I believe there are 2 boxes that are used to calculate it. I think it's, oh, I don't have it off the top of my head. If you please send me this one to answer by text. There are 2 boxes that determine the shift value. It's, I think, number of, of proteins per asymmetric units and or like number of asymmetric units, and then I think the rise determines the shift in Reliant. And sometimes we set it as, we control it by setting one of them as 1 and the other one to the desired value. And so that will determine shift. The other question was why straight filaments. The alignment into the very center of the box, straight filaments, is really important for subsequent 3D classification because the sintering of those particles during extraction might be pretty poor. And with poor sintering, it's just going to get some artifactual results or just not contribute to the signal. And so getting really good sintering before re-extracting the particles to get really nicely sintered boxes is pretty important.
Next question is, could you comment on salt concentration in samples for grid preparation and imaging? What was the highest salt concentration allowed that gave you good images?
Yeah, so we've never tried it to where we have very high salt. We're fortunate where everything behaves pretty well at 150 millimolar. And, I was very weary of this as well. So, like I mentioned earlier, we dialyzed after anion exchange in order to step down the salt concentration to 150. I'm in lab, I don't know if we've had anyone do salt concentrations too much higher than that. And when salt components are introduced by gradient ultracentrifugation or other methods, generally people like to desalt before going on the cryo-EM grid. So I would, maybe I could find a reference that suggests maybe what the maximum is, but we like to keep it as low as possible.
This is the last question we're probably going to get in, but I have had a hard time using PEG for transfections. Have you tried, or sorry, PEI? Have you tried PEI? I can tell you that name, but I'm going to slaughter it. Let me know. It is cheaper than the system that you mentioned.
Yes, so we use PEI to transfect in the XP system. So we don't use very expensive transfection reagent. There's things like FectoPro that have potentially higher transfection efficiencies. That comes at the cost of they're physically more expensive. And for some proteins, we found that higher transfection efficiencies actually lead to more aggregation in the cells, and then we can't purify a good amount of protein. And so for expression, varying the amount of plasmid transfected with PEI or FectoPro, or trying different temperatures like 30 degrees or 37 degrees for mammalian cell transfection is really important, and then doing a time course expression. So all of these things can be very important. For my project, we do use PEI. I have the protocols.io posted on it's available now. And so I'd say go check that out because I just go into pretty much everything having to do with the PEI transfection. I think maybe I even mentioned how we make the PEI in-house or link to a source. And so there you can read exactly how I did it. And if you have questions, feel free to just email me. I think my email is somewhere on the internet. And so feel free to reach out.
All right, we have 1 more minute. Do you want to answer one more question? Or should we end it?
Sure, one more question then.
Okay.
That's great. I'll get some water.
So I recommend you use the osmolite TMAO in order to stabilize your complex. We saw this by the thermal shift assay. It's more of a statement than a question.
I'll write that down. I'll look into it. That's very interesting. I haven't heard of it, so I'd love to take a look.
All right.
Awesome.
Looks like there's going to be 8 questions for you to answer after.
That was a fantastic information overload, Bobby. I think we touched on so many issues. So thank you so much for coming and providing your expertise. And it's really awesome that so many of your resources are just freely available. So hopefully any unanswered questions are easy to answer using the resources you've shared. Thank you so much.
And I think right now, in the absence of in-person interaction because of COVID, it's super important to make this stuff as available as possible. And just know that, it's great to get an email from somebody, and if I could provide something that helps, that's fantastic, because myself, I've had so many good tips. And so feel free to email at any time, especially because we're not bumping into each other now at conferences.
All right, and for those of you who hung out this long, we're very glad you did. We assume that means you find this useful, and we're very happy about that. And we will see you hopefully next month. Bye.